Thank you everyone for joining us today and welcome. So today we're going to be going over some more advanced features of the BaseSpace Sequence Hub command line tool. I'll be referring to it as BaseSpace CLI or just CLI, just so you know. So our agenda for today, we're just going to do a quick recap of things you should have learned and seen in the Part 1 webinar for BaseSpace CLI. Then we're going to go through a few demos. We're going to interact with data from different work groups, show you how you can do that. Then we're going to jump into uploading non-FASTQ files. So uploading FASTQ files is one thing covered in Part 1. It is possible to upload non-FASTQ files for other types of analyses and supporting files for those analyses. We'll go through that. And finally, we're going to go through an actual demo where we're launching analysis applications directly from the command line using our FASTQ files as well as our non-FASTQ files. So just as a quick recap, these are some of the lessons that were taught in the Part 1 webinar for BaseSpace CLI. The first and foremost was downloading and installing the actual program and then authorizing it to work with your particular work group or your BaseSpace account. The next was uploading FASTQ files so you can actually process the data and do analyses with the apps on BaseSpace. And then we also went over downloading run and project data from BaseSpace. So I will say here that the format of the commands I'm showing are specific to the Windows or DOS command line. For the most part, the command structure is identical for the Linux and Mac OS versions, but you'll have just a BS instead of the bs.exe. That .exe is specific to the Windows executable. There is a command later that is an example of this, and I'll call that out when we get to it. And the format of this webinar will have, before each of our demos, we'll have a slide like this where I give you a preview of the commands we're going to use, and I've broken them up here based on the type of item in the command line. So down at the bottom, I've defined what the colors mean. So the bs.exe, or if Linux and Mac OS, it'd just be BS, that's the actual CLI program. The things in bold blue there are the command and the entity that the command is being run on. The things in green are the CLI options, so options that the blue command expects or can ingest. The things in orange are the arguments that are passed to those options that are then brought to the BaseSpace CLI command. And then files that we're working with, uploading or downloading, are going to be in red just to separate them out from everything else. So the first thing we're going to get into, and this is a bit of a recap from Part 1, is just interacting with different work groups and authorizing your CLI program to interact with both your personal and professional work group. So the first thing that we can do is authorize our BaseSpace CLI program to interact with a professional work group. In this case, I'm just calling it Pro, and the --config option is telling the BaseSpace CLI to create a configuration file called Pro that will contain the authentication token specific to the work group that you want to work in. So what this does is generates an authentication link. When we go over to the BaseSpace website, here we're actually signed in under our professional work group, and it brings up this authorization page, and it lists the different permissions that we have for interacting with this particular work group. And once we hit accept, we have now authorized our CLI program to connect to that work group. And we have to be certain that when we want to interact with that specific work group, that we always use the correct config file and call it out with that config option as seen here. And just to show you, we've run a whoami command, and that just says what the name of the account that you're logging into, or the work group in this case, and then it has the scopes listed, which are another way of saying permissions that you have when interacting with that work group. And the scopes are important, and they'll come into play later. And one thing we can do is update those scopes, essentially overwrite our configuration file with added permissions, or we can actually remove permissions if we want to. So this next example just shows us how we can overwrite that pro config file we made. And to do so, we're going to be using the force option, which is designated as --force. This is telling CLI to overwrite the original pro config file. And here we've added a scope called manage applications. So everything else that's in that scopes list is already there up above, but we've just added the manage applications, and this will come into play later when we actually try to interact with and launch applications on BaseSpace. So if you don't provide the force option, you'll get an error saying that the pro config file already exists. So the force is necessary to overwrite that. Then it generates, again, a new authentication URL. So when we go over and we're logged into that same work group, we can just go ahead and sign in. It will pull up that authentication page, and it will list the permissions. And just to double check, we're going to scroll down, and we see manage applications has been added. So we hit accept. And now we should have authorization to manage applications through that professional work group. Just to double check, we're going to run that same whoami command, and we're going to see if indeed the manage applications scope has been added. So for the remainder of this webinar, I have actually made my default configuration file to this professional work group using that force command to overwrite my original default. And you can do this so that you don't have to constantly type in the config pro or config workgroup name option, just to make your life a little bit easier so you don't have to always remember it. So that's what I'll be doing going forward. And again, if you want to overwrite a command, or sorry, overwrite your config file, you can always use the --force. All right, so in our first demo, we're going to get into uploading non-FASTQ files. And we're going to do this in the context of a DRAGENT example. So DRAGENT is one of the suite of analysis software that we have available on BaseSpace. And in this case, we're going to upload a custom genome in the form of the generic FASTA or FASTA file. And we're going to manipulate that file using one of the DRAGENT apps. And that is because DRAGENT requires building of what's called a hash table for a genome. And this is a thing that the DRAGENT software converts the FASTA file into a hash table that it can understand and use for any actual downstream analyses, such as our germline or somatic pipelines. So we're going to do this before we actually get into using one of the DRAGENT applications to run an actual analysis. So here's just a preview of what we're going to use for this segment. First, we're going to create a project up on our BaseSpace account. We're going to upload our non-FASTQ file. In this case, it is a FASTA file. I said custom genome. In this case, it's just the MM10 genome, just as an example, but it can be your custom genome that you desire. Next, we're going to retrieve the new app result from that upload session. And this is important because we need it to then retrieve the file ID for that newly uploaded FASTA file. So every file that you upload or save to BaseSpace will have a unique file ID. And in our demo today, we will need those file IDs to specifically link to those files. So let's do the first step, and we're going to create a project. And this command is pretty straightforward. It's create project, just like it sounds. And the -n specifies the name, and the argument we're passing into it is DRAGENT alignment. So that happens pretty quickly. We can see the name is DRAGENT alignment as we specified in the command. And it's also provided us with a project ID, which we'll use going forward. So the next step is that we actually want to upload our data set. Now, we're not going to be uploading a FASTQ file, and the default file type that the CLI expects when uploading is a FASTQ file. But in this case, we're just going to specify that it is not actually a FASTQ file. So we're going to use the upload dataset command. We're going to point it to our project, that new ID we just copied from above. We're going to give it a name. This is just the name of the upload session. And I'm going to call it MM10FASTA. And then we're going to point to our actual file in the folder we're working from. And the last option is most important. We actually need to specify the type with the -t command. And if you use common.files, the CLI program knows that this is not a FASTQ file and does not check it as though it were one. So it uploads, and once it goes to 100%, it'll check it. If it uploads complete, it'll let you know. And now we can see that our file is actually uploaded. And to double check that, we can use the list app result command. And what this will do is after that app session, the upload app session is complete, we can list any app results in that same project that we uploaded to. And we'll see here the name MM10FASTA that we gave it, and it's got a new ID that we can use. Now we need this ID. This ID is specific to the app session, not the file we uploaded. So it's possible to upload multiple files into an app result, they will be contained within the same app result, but they will all have unique file IDs. So one more step is required to grab that file ID, which we're going to need when we go into the next step. And that's the content command. So we're going to run the content command on that app result. And we do that by specifying content, then app result. And the input is going to be the ID of that new app result. When we run that, we see, indeed, we have our mm10.fa, which is the same file name as that we uploaded in the 2 steps previously. And we now see that we have an ID, and this is the file ID that we're going to need to copy and use later. So now that we've uploaded our non-FASTQ file, in this case a FASTA file, we're going to actually do something with it. So we've completed these 3 steps. The next is to actually build that reference hash table that DRAGENT requires for its downstream applications. And then we're actually going to run a downstream application, in this case the germline pipeline, using that newly created hash table from our custom FASTA genome. Okay, so for this section, we're going to go through listing applications. We're going to go through finding out what options are available for those applications on BaseSpace, as well as which ones are required and optional. The next thing we're going to do is actually launch the reference builder, and this is the program that takes our MM10FASTA in this case and creates a hash table, a whole new file that DRAGENT can understand and use for the germline pipeline. And again, as we did before, we'll need to get the file ID for that newly generated hash table using the content app result command. And our final command, kind of the culmination of everything, is that we are going to launch the DRAGENT germline pipeline using our newly created hash table and some FASTQ files that we have previously uploaded. And you'll see here that I've highlighted some things in purple, and these are app options. So these are options that are passed to the BaseSpace application itself, not to the BaseSpace command line interface tool. And we'll go over a little bit more of how to properly specify those. Okay, so here the first thing we want to do is list our applications. Let's see what BaseSpace has to offer. So this is a big list. It's all of the applications available. And you may not want to search through all of these if you have a lot of applications listed. But what you can see is it gives a name, an ID, and a version number. And they're all listed and kind of spread out. So one thing we can do to hone in on the applications we're interested in, especially if we know something that's in the title or the name of the application, is we can actually filter based on a particular string. In this case, I'm just looking for the word dragon. And this command, the find str or find string, is specific to Windows. And what it's doing is taking the output from the list application command and then looking for the word dragon. So this similar command can be run on Linux or Mac OS, and that's the grep command. Many may be familiar with this tool. It essentially does the same thing. It looks for a string within either a file or an output. And you'll see here I've shown that the bs.exe for Windows and bs for Linux and Mac. This is just because Windows has the .exe executable, and it may be required to include that when running from the Windows command line. From the Linux and Mac OS, the command there is what you would do to get the same output you see up on the terminal screen there. All right, so now we have a list of our Dragon applications. We have their names and IDs. We can interact with them with using the name or ID, and that's a personal choice for whoever's building the tools that they want. In this case, I'm going to use the name just so it's obvious which one which application we're actually looking at. So in this case, I know it says launch application, but I'm going to add the list option. And what this does is lists all of the options available for that application on BaseSpace. So all the way on the left, we have the option name, and this is how we'll specify what value we want for that option. Then we have some choices that we can choose from, what the defaults are, and then all the way on the right, we have whether it's required or not. So we can see here that there are 3 required options for this command. That is that we have to give it an app session name, we have to tell it what project to save to with the project ID, and then we have to give it an actual file ID for the FASTA file that we're using. So let's actually launch this, and I'm going to go through an example of how I like to do things, as well as show you another way to output that same information in a different text format. So we run that same command, but we're going to tell the format to be a CSV or comma-separated value. And this is sometimes easier for parsing if you're doing things like automation. It lists the same information. You can see it has the same header information, the option, and then whether they're required all the way on the right. So what I'm going to do is I like to build my commands in a text editor and check everything before I launch it. So I'm just going to use the 3 required options here. I'm going to copy and paste them. And you designate the value for that option with a colon and then no spaces between the option and the colon. So here we have app session name, colon, and I'm saying mm10 ref build. The quotes you can use if you have spaces. I used them here, although they wouldn't be necessary because we don't have any spaces. Then we have to give it a project ID, which is going to be our same project ID from before. And finally, the FASTA ID that we got from the content app result command earlier. And you'll see here that all of these options that are being passed into the actual Dragon application are preceded with a -O. That tells the CLI program to take that option and pass it into the BaseSpace application. So we paste that in, it looks good, we hit go, and when it launches the application, we should get a name, we'll get a session ID, and then we'll also see the execution status, and because we just started it, it's in initializing. So this is going to build our hash table from our FASTA file, and it may take a while, so we're going to go over some of the next commands we're going to use that we kind of have done in the background to build our final application launch command. So again, we're going to list the app results for our project. We did this exact same command earlier, but this time we're going to have another app result when the Dragon reference builder is done. So we'll list that. and it'll give us a new ID, which is shown here. We will retrieve the specific file ID within that app result, and that's going to be the hash table that we've created. And we'll need that file ID to then pass into the DRAGENT germline pipeline. And lastly, a cool little tool is that we can actually check the status of our app sessions, and we can specify how new or old we're looking for, the time range that we want to look at, to just check on our app session statuses without having to actually go into BaseSpace on the web. We can check it directly from the command line. All right, so our final command here is going to be for the DRAGENT germline pipeline. So we're going to launch that application. I'm going to specify by name again, just to make it easier to follow along. And then we're going to give it an app session name. In this case, I'm just going to call it the MM10 germline run. And because I want to save it to the same project, I'm just going to give it that same project ID. It's the same option, project-ID. And then we're going to tell it what type of hash table we have with the ht-ref. In this case, it's a custom. V7 just tells it what version of DRAGENT hash table we're using. In this case, we need to use version 7. And then here, the ht-ID is the actual file ID that we retrieved using that content app result command. And lastly, we need to give it some samples to actually run it on. And these are some previously uploaded samples, and here I'm specifying the biosample ID for our mouse whole genome sequencing that we uploaded previously. Once that's all done, we'll hit enter, and it will pop up our status. And again, you can see it's got its own ID, and it says it's in initializing. So what we can do is use that very last command I had on the previous slide, and we can actually just check the status of our most recent app sessions. And here, I'm just going to list the app sessions that are newer than 5 minutes. So if you do that, then only those that are 5 minutes or younger will be listed, and we can see here in the execution status that it actually went from initializing into pending execution, which is one step closer to running. And just to double check everything, we're actually going to hop over to our BaseSpace account, and I'm just going to refresh the page, and we should see that that same app session is here under the Analyses tab. And indeed, we have our germline pipeline. It's in pending execution, so we can let that run. So that's kind of a culmination of how we can upload non-FASTQ files that may be necessary for certain applications. This can apply to things like manifests and embed files. We can run applications using those after we find the file ID, and then we can pass the results from those applications into further downstream analyses, as we've done here. And just for convenience, I've listed out all the commands we use throughout this presentation and a brief description of what they're doing, just as a kind of a nice little cheat sheet if anyone wants to try running these with any of their data.